Hello, everyone, and welcome to today's webinar titled Savor the Flavor, Keeping Meat Products Safe with a Veterinary Drug Screening Comprehensive Workflow Solution. I am Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Agilent. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you like at any time you like during the presentation. To do so, simply type them into the Ask a Question box and click Send. Please address each question to each specific speaker. We will answer as many of your questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing the presentation, click on that Ask a Question box located on the far left of your screen. I would now like to present today's speakers. Dr. Patrick Batoon, Product Manager of Quadrupole LC, and Siji Joseph, Solution Scientist, both of Agilent Technologies. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. Gentlemen, welcome. You may now begin your presentation.
Hello, everyone, and thank you for attending. This talk is called Savor the Flavor, Keeping Meat Products Safe with a Vet Drug Screening Workflow Solution. Accompanying me in this talk is my colleague, Siji Joseph, who is an application solution scientist, and we're here to talk about routine veterinary drug analysis and to promote our solution for the challenges faced within this field. To set the stage for this topic, the first question to ask are, what are veterinary drugs, and why do we want to monitor veterinary drugs in food? First off, veterinary drugs are extensively and commonly used to improve the growth and health outcomes of livestock farm animals. This prevents the transfer of parasites and other diseases used to treat physical ailments like cuts, scrapes, and bruises, or to even increase growth or secondary product yield. These drugs elicit some kind of physiological response. Many of these vet drugs are the same medications used to treat humans, thus improper use can result in some serious adverse effects, acute, immediately, or chronic, built up over time. Thankfully, many governing bodies require the monitoring and reporting of residue levels for the various primary and secondary food products from these animals. Some examples of vet drug treatments are for the treatment or prophylaxis of parasites and cellular diseases. Anti-helminthic drugs are used to treat parasitic worms, like the deworming medication you give to your household cat or dog, or coccidiostats, which are used to treat coccidioidosis, which is an infection by some kind of parasitic prokaryote and can be highly infectious and damaging to a livestock herd. Additionally, some vet drugs are not vet drugs at all, as they are the standard medications used to treat humans. These can be common NSAID painkillers like paracetamol, ibuprofen, aspirin, or in humans, prescription-grade corticosteroids. And lastly, sedatives or tranquilizers used to calm the animal down, such as in the case of giving birth or during animal transport. Some compound names you may have heard of are alprazolam, otherwise known as Xanax, diazepam, Valium, or ketamine. Regardless, these vet drugs were made to elicit some kind of physiological response, and when transferred to the consumer, can cause very adverse reactions. As mentioned earlier, the presence of vet drugs is highly regulated because effects can easily be transferred to the consumer. However, various regulatory bodies set their own maximum residue limit, or MRL guidelines, that differ animal to animal, country by country, or region by region. One of the stricter examples are regulations by the EU, which has defined a forbidden compounds list in food, which may or may not be adopted by other parts of the world and are subject to evaluation by those other countries or regions' own experts. In other words, the regulatory landscape for vet drugs is highly complex because various drug classes are active at various physiological levels, and what is acceptable to one country may be rejected by another. The term maximum residue limit, or MRL, is applied to the analysis of all foods and not limited to meat or dairy. In the case of screening for pesticide residues in fruits and vegetables, however, there are generally two MRL targets to consider. Zero residue, meaning nothing is allowed to be detected, or a general benchmark of 10 ppb, regardless of the plant matrix. Vet drugs, on the other hand, are regulated to be zero residue, or of various MRL levels depending on the animal, chicken, beef, pork, shrimp, etc., or the matrix type, dairy, muscle, fat, organ. And this is because, as mentioned before, the vet drug activity and concentration will depend on class. So a simple 10 ppb MRL, such as that used in pesticide analysis in fruits and vegetables, may not be feasible. It is entirely possible that a vet drug exhibits some kind of effect at the 10 ppb level, so a lower MRL value may need to be defined. Since there are so many regulations and guidelines establishing MRLs for vet drugs, MRL varies based on different region and different matrices. This makes the comparison to pesticides highly complex for vet drug analysis. Some countries utilize the 10 ppb pesticide guideline, but because vet drugs have different physiological activity levels, they vary region by region. Based on the points made in the previous slide, you can get a sense of the complexity faced when monitoring for vet drugs. Maximum residue limits vary depending on geographic region, the type of matrix, or the classes of compounds. People interested in monitoring vet drugs need a robust set of instrumentation and a robust workflow, but also able to reach the sensitivity limits required from these regulations. As you can imagine, grinding up some animal meat or diluting some egg or milk can easily soil the instrument, and the steps to remedy this can be extremely time-consuming or challenging. Lastly, you can have the best sample extraction materials or the best laboratory instrument in the world, but still face challenges, especially in the case of having the skill to develop efficient sample preparation or instrument workflows. And hopefully, by the end of this talk, we've demonstrated that we've taken many of these considerations in mind to develop an easy-to-use, streamlined veterinary drug workflow that is sensitive enough to reach the most rigorous regulatory limits in a consistent and reliable manner. A major problem faced in the field of vet drug analysis are that these drugs are highly heterogeneous in regards to chemical structure or mode of action. Oftentimes, these workflows are split logically in some way that is convenient for the lab. For example, the analysis of a specific chemical class may need specific chemistry, specific sample prep to produce multiple extractions from a single sample. Or another way to approach this is to run multiple chromatography mass spectrometry methods from a single extraction. Either way, this is a major problem for the lab because there may be redundant steps involved, being repeated multiple times. If we just consider the examples on the side, for example, the detection of antibiotics, antihelmintics, and NSAIDs are done separately, it will take about 1 hour per analysis for a single sample. This essentially means that resources are consumed for each analysis and is highly throughput limited by time. Our solution that we're promoting today is a multi-residue approach, start to finish, otherwise known as end-to-end, meaning that we want to consolidate the multiple analysis of a sample to a single analysis that can detect and quantify the various compound classes simultaneously. Based on the example shown previously, this will take in at least 60-minute instrumental analysis to something that takes about 15 minutes, keeping in mind that this does not include the sample preparation steps, which further lengthen the workflow. And lastly, things to think about how to actually do this is how do you streamline the process so it's simple, efficient, and scalable so that we can be providing the lab scientists something of value compared to if they develop the workflow completely on their own. So as I transfer my presentation over to Siji, the applications and workflow scientist, I just want to point out the challenges to developing a streamlined multi-class workflow method. For our primary considerations, we aim to develop a generic sample preparation method that can be applied to many sample matrices, beef, chicken, pork, etc., and encompass the wide array of chemical classes within the scope of the veterinary drug umbrella. To complement the generic sample prep must be a generic instrument analysis method, maximizing the full potential of the chromatography and mass spectrometry system. The method must have high sensitivity to meet the regulatory targets, while also eliminating false positives or false negatives. Then to ensure that this workflow is truly valuable for its users, we have these secondary considerations like robust for day-to-day operations, that the workflow is cost-effective by maximizing the utility of each component, the consumables, instrument, reagents, etc., and lastly, that it is scalable to address a large volume of samples. So now that I've laid out the challenges, I would like to transfer over to scientist Siji Joseph for talking about how he and his team have addressed many of these challenges in a veterinary drug workflow.
Thank you, Patrick, and hello, everyone. Before going to the technical updates, let me share the instrument configuration and some of the key components in the flow path. We used a 1290 Infinity 2 LC system coupled with Agilent triple quadrupole and Agilent consumables to perform the workflow. A high-speed binary pump, a multi-sampler, and a multi-column thermostat are the individual modules of the LC stack. A high-speed binary pump is an ideal front end of the high-speed LC-MS applications. The multi-sampler is a high-capacity injector module, enables to handle the increased number of sample load and best suitable for precise injections. The needle assembly was washed with 3 solvents using the multi-wash feature of the module, and thus minimized the carryover issues. Inline filter included in the multi-sampler flow path helped to increase the column life while handling complex matrices. The multi-column thermostat connected with Infinity Lab PowerShell 120C18 column helped to perform the fast gradient and ensured day-to-day reproducibility. The workflow performance was verified using 2 models of Agilent triple quad systems, 6470B and 6495C. These robust triple quad systems, together with Massander software, helped for reliable and sensitive target detection using DMRM acquisition mode. The Agilent JetStream ESI source and WalkShield features help to collect the uninterrupted data from a wide range of matrices and increased lab productivity. Agilent also provides a set of consumables which ensures the day-to-day reproducibility of the workflow solution. The Captiva EMR Lipid cartridges included in the consumable list is very selective and efficient for lipid removal without any analyte loss. The bundled consumables ensures the confidence in sample prep reproducibility. The overall workflow summary is shown in this slide. 2-gram sample in a 50 ml polypropylene tube is the start point of sample prep. VetDrugs standards were spiked to this 2-gram sample to get pre-spiked QC samples. In this workflow, we used 3 different concentrations of QC samples to evaluate recovery. The QC sample concentrations are spread across a wide calibration range. Replicates of QC samples were also prepared to verify the recovery repeatability and reproducibility parameters. The unspiked sample was used as a blank matrix. Both spiked and unspiked sample tubes will go through the same sample prep protocol, which is a liquid-liquid extraction followed by Captiva EMR Lipid cleanup. The 3 different concentrations of matrix spiked extracts will be further diluted to prepare ready to inject low, mid, and high QC sample vials. The unspiked sample tube will eventually result in a blank matrix extract, and this blank matrix extract will be used to prepare matrix matched calibration levels and matrix blank vials for LCMS analysis. Now let's go through the various steps involved in sample prep protocol. The workflow is verified for chicken, beef, and pork muscle matrices. The first step is to weigh finely minced 2-gram muscle sample into a 50 ml polypropylene tube. For simplicity, I will guide through the various sample prep steps using one each of spiked and unspiked sample tubes. Spike the veterinary drug standards to one of the tube for matrix spiked QC. Vortex the tube for 30 seconds to mix the standard well with the sample, then wait for 20 minutes to infiltrate the standard into the sample matrix. Keep the second tube as unspiked for matrix blank. Next is a two-step liquid-liquid extraction. This includes the first extraction with aqueous buffer, followed by extraction using acidified organic solvent. A mechanical shaker is used to automate the extraction procedure, followed by a centrifugation step to settle the sediments and get a clear layer of the extract. Next is Captiva cleanup. Captiva EMR Lipid 3 ml cartridges together with positive pressure manifolds are used to clean up the extract. Performing Captiva cleanup is very easy, and we don't have to do any traditional SP steps such as preconditioning, conditioning, washing, etc. The Captiva cartridges are designed for gravity elution and allows a hands-free operation once the sample is loaded onto the Captiva. First, pass the extract through the Captiva cartridge, and during this step, lipids are trapped in the solvent while target analytes are passed through the cartridges. Then perform a second elution using 90% acetonitrile to maximize the target recovery. Homogenized eluent from the spiked sample is used to make the final ready to inject QC samples. The blank matrix extract from unspiked sample is used to prepare ready to inject matrix blank vial and post-spike calibration levels. This slide summarizes the performance criteria we used to demonstrate the success of the newly developed VetDrugs solution. We are very confident in the overall performance of the VetDrugs solution because of the extensive tests we performed while developing and verifying the analytical protocol. The targets are identified from AOSC guideline and EU-US regulations, and verified the methodology by following standard method performance guidelines from AOAC. The test includes sensitivity, which includes LOD and LOQ, and linearity study using matrix matched calibration levels. The recovery evaluation was performed using 3 matrix spiked QC samples. Also, intraday and interday recovery analysis using matrix spiked QC samples. Next several slides, we will show you the results generated with Agilent 6470B triple quad using chicken muscle, and conclude the presentation with a comparison study using beef and pork muscle. Is to showcase the MRM overlay of 210 veterinary drugs, post spiked at 2.5 microgram per liter in the blank chicken matrix. The symmetric sharp peak shapes demonstrates the efficient chromatographic separation of targets within the 13 minutes elution time. 2 or 3 target-specific MRM transitions are included for each compound, and the MRM transitions are optimized using MassHunter MRM Optimizer. The insert plot is a zoom-in view of normalized peaks corresponding to 6 early eluting targets. The sensitivity is always a high-demand analytical measure. Limit of detection, LOD, is one of the important criteria, and LOD data of all 210 targets are summarized in this bar chart. LOD of all targets are less than 10 ppb. You can see from the chart, LOD of majority of the targets are less than 1 ppb. In addition to the sensitivity bar chart, The slide included two overlay MRM traces as example data. The first is overlay of MRM duplicate injections of sarafloxacin VetDrug at 0.25 ppb. Signal to noise is the important criterion to establish LOD. But here, in addition to signal to noise, we have also included filters like closeness of ion ratio, increased area compared to blank matrix, area RST from triplicate injections to assign 0.25 ppb as LOD for sarafloxacin. The right side trace is an overlay of MRM for the same target at 0.5 ppb. Here also, we verified all these performance characteristics of the peak to label as limit of quantitation, LOQ. This slide summarizes the highlights of dynamic MRM mode. Agilent triple quad system enables to perform sensitive detection of hundreds of targets simultaneously. The DMRM mode allows the user to input a fixed cycle time, and the software automatically adjusts the MRM dwell time for a specific time period. Flexibility to add hundreds of MRMs within a method, but allows the instrument to do the active MRM monitoring at the relevant time. Changing the cycle time will not significantly compromise the quality of MRM data. Customers can still add more MRMs to the existing method. Good linearity is a basic need for any quantitation-based analysis approach. A good sample prep protocol means consistent reproducibility of linearity across different matrices. As a part of extensive inter-batch studies, many calibration curves are generated for all targets. An overlay of linearity curves for praziquantel is shown in this slide as an example. The calibration curves are using matrix-matched spike samples from LOQ, which is 0.25 ppb, to 100 ppb. The overlay shows that all calibration curves have consistent R-square values and slopes. Please note, these all done in different batches, either different batches of chicken muscle or beef or pork muscle. This illustrates the efficient lipid removal of Captiva EMR cartridges, regardless of fat distribution across food matrices. The instrument precision and accuracy results are also excellent. The data shown here is RT precision and accuracy results generated using matrix-matched calibration levels in chicken matrix. The left chart is of RT precision, and the RT RSD values are way lower than 0.5%. The RT precision RSD results confirm the reproducibility of LC elution profile and column performance. The average accuracy values were calculated for each post-spike calibration levels, and the values of all targets were within the limit of 70 to 120%. The example data shown here at the right side is for 10 ppb level. Recovery is another criteria for method performance evaluation. More than 97% of targets demonstrated good recovery from 60 to 120% in chicken matrix. This is an excellent result if we consider a single sample prep for multi-class VetDrugs. Recovery was calculated using target response in matrix spiked QCs and measure response using matrix matched calibration curve equations. The slide shows an MRM chromatograph overlay of the three targets, trimethoprim, oxybendazole, and febantel. The post-extraction sample trace is in black, and the pre-extraction sample trace in blue, at a concentration corresponding to 1 microgram per kilogram in chicken. The response counts comparison between post-extraction and pre-extraction QC samples indicates a good recovery, approximately 100% for all these three targets. While developing and verifying the solution performance, we did extensive intraday recovery repeatability and interday recovery reproducibility to ensure the reliable day-to-day operation of the solution. The interday reproducibility study was performed with different sample loads, different analysts, different instruments, different laboratory environments, performed on different dates. The plot at right side shows inter-day recovery reproducibility results generated using chicken matrix. All results of all the targets falls within AOC limit of 32% RSD. Also, please note, inter-day recovery reproducibility of more than 91% of the targets are within a tight RSD of 15%. The comprehensive veterinary drug DMRM solution supports the analysis of beef and pork muscles also. The data shows here a 10 microgram per kilogram middle QC sample recovery of beef and pork muscle. The beef data is on the left side and the pork data is on the right side. The recovery results are in good agreement with that of chicken matrix. More than 97% of the targets were within the acceptable range of 60 to 120 in chicken matrix, while more than 94% of targets were within the acceptable range of 60 to 120% in beef and pork muscle. This confirms the applicability of the workflow solution in other matrices also. We did evaluate the matrix effects as well. Like any other reliable protocol, veterinary drug analysis in food matrices is also very challenging. Not all targets are well behaving. Some are giving real trouble in matrix. We did a comparison between neat standards with matrix matched spikes to evaluate the matrix effect. In all three matrices, more than 93% targets well behaved and not having significant ion suppression. 4% of the targets demonstrate some average level of ion suppression, while 3% of targets experience significant ion suppression. On the right side, we are showing three example situations. The blue is neat standard, green trace is post-spike, and the red trace is pre-spike. The first example is with trimethoprim, where all traces are the same intensity. We can say that the target trimethoprim did not exhibit any ion suppression, and the recovery results are very good in this case. While in the case of sulfisomidine, observed high ion suppression, but recovery results are satisfactory. Example situation is for the target erythromycin. This target also experienced high ion suppression, and recovery results are also suffering for this target. Details of all this data are included in the Agilent application note 5994-1932EN. A DMRM database is also included in the Agilent Comprehensive Veterinary Drug Workflow Solution. The database includes details of all 210 targets and all relevant MS details. We can filter the targets in various ways, like regulation, chemical classes, or CAS numbers. The database can be used to customize acquisition methods for a set of interested targets in a convenient way as per local regulatory requirements or laboratory needs. This slide summarizes the technical results of workflow. The results summarized in 10 bullet points. The Captiva EMR Lipid-based sample prep approach is like a hands-free sample prep. It can parallelly process up to 48 samples in less than 30 minutes. The column included in the workflow solution helped to develop faster gradient for multi-class sensitive LCMS analysis. The workflow covers 210 targets from 28 different chemical classes, and these targets are listed in AOC, EU, and US regulations or guidelines. The LOD of all targets are ranging from 0.1 to 10 ppb, and the sensitivity meets the MRL screening requirements. Precision and accuracy of all the targets were also evaluated and is within the acceptable limits. The linearity of all targets evaluated using matrix matched calibration levels from LOQ to 100 ppb, and R-square values are above 0.99. The recovery analysis was also performed using 3 matrix spiked QC samples intraday recovery repeatability, and inter-day recovery reproducibility were also evaluated to confirm method robustness. The MRM database included in the workflow solution helps to customize the methods based on local laboratory needs and regulatory requirements. Applicability verified for 3 matrices like chicken, beef, and pork. Workflow performance verified using 2 models of Agilent triple quad, 6470B and 6495C. The solution also includes system suitability standard mix for continuous monitoring of the solution. The standard mix is a cocktail of 25 VetDrugs standards and covers 10 different chemical classes and favoring both positive and negative mode ionization. A typical resulting DMRM chromatogram of the test mix is shown here. We can use this standard mix as a QC sample for routine monitoring of the solution to ensure confident day-to-day operation. Besides, the system's suitability test mix can also use to perform any troubleshooting or performance checking of any tests like linearity, carryover, inter-day repeatability, etc. A customized mass under report template included in the solution helps to quickly summarize the performance evaluation and quality check report with the system suitability test mix. Both the test mix and the custom report helps the laboratory to implement in-house lab operating procedures to monitor the performance of the workflow and thus prevent any unplanned downtime. Thank you, everyone. And now Patrick will guide through remaining slides and will share a few other exciting product-related information. Patrick, over to you.
Thanks, Siji, for the great technical details described. Moving on to the actual workflow product, we've managed to package it into a kit which includes the consumables, columns, method parameters, reporting templates, and other supporting material. If you're interested, you can check out the fully public and detailed application note for all the information provided, so you can develop methods on your own. However, the nice thing about the kit product that we are promoting is that it is a complete package of everything that is needed for successful execution of this particular workflow. In regards to the regulatory environment, we cover at least 200 analytes, which fall within the regulatory jurisdiction of the various organizations here, with the distribution of chemical coverage shown in the pie chart. We've chosen these regulatory organizations because they've provided the most rigorous MRL guidance that were built into our methods. Again, there are a variety of technical references you can refer to if you're interested and would like more information. Otherwise, feel free to reach out to your local Agilent sales rep or go through our website and have someone contact you, and we can get you the pertinent material as soon as possible. Just to give a broad overview of Agilent's triple quadrupole LC-MS portfolio, we only have 3 instruments serving different aspects of the routine workflow. In regards to the comprehensive veterinary drug solution, it is a quite stressful method, so we take advantage of the research-enabled aspects of the 6470 and the 6495 LCTQ. In terms of product launches this June 2020, we have a new revision of the 6470, which we refer to as the 6470B, adding many of the newer functionalities from Ultivo and the 6495C, such as VacShield. At this point, all of Agilent's LC-MS triple quadrupole instrumentation are VacShield enabled to maximize the instrument uptime for routine applications. To close with some final notes, a comprehensive workflow was developed to analyze multiple classes of veterinary drugs simultaneously. This workflow covers at least 200 analytes and is fully validated on the 6470B, 6470A, and the 6495C LC-MS triple quadrupole. This multi-class workflow was done to consolidate several single-class methods. The workflow described was developed into an actual comprehensive end-to-end solution product that was fully validated cross-lab and cross-instrument for the models described. The workflow solution was designed to be end-to-end, covering sample prep, instrument analysis, data analysis, and reporting for users of this kit to successfully execute veterinary drug analysis in a scalable manner. Lastly, this workflow kit can be customized to local laboratory and regulatory requirements to suit our users' needs. So with that, feel free to reach out by consulting your local Agilent representative, visit our website, or feel free to search online for the reference material we've described. And with that, I'd like to open the floor to any questions or comments that we can try to address. Thank you very much.
Thank you, Patrick, and thank you, Siji, for your informative presentation. We will now start our Q&A portion of the webinar. Now, to our audience, again, if you have any questions you would like to ask, please do so now. Just click on that Ask a Question box on the left and click Send. Now, let's get started. Okay, gentlemen, let's start with our first question here. Could you please elaborate on what was meant by method customization?
Hello, can you hear me?
Yes.
Thank you for the question. Yeah, what we meant by method customization is like, even though we have a comprehensive method addressing 210 targets, maybe a user interested target may be different. They may be focused only on targets specific to chicken or specific to beef or maybe specific to EU regulations. So what we meant by method customization is like we are giving a database, the MRM database. With that database, we can easily customize a method, MRM method, with the targets interesting to the local laboratory need or regulatory requirements. So it's very easy to search targets from this 210 list and create a subset method very easily with few clicks. Thank you.
Thank you, Siji. Now our next question. Is the kit instrument specific and can it be used in other MS instruments? Siji or Patrick?
Can you please repeat the question one more time, please? Thank you.
Yeah, absolutely. Is the kit instrument specific and can it be used in other MS instruments?
Yes, so right now the verification is for 2 models. Of course, we don't have the data with other models, but it can be adopted to other models. Yeah, thank you.
Thank you.
I just want to add.
Oh, Patrick,
sorry, go ahead. Yeah. I just want to add that the results presented and the kit is kind of instrument specific in that the methods that we show and presented are what is successfully done with the 2 models. If you attempt to try this on another instrument model that we didn't present for, we can't really guarantee your success.
Thank you, Patrick. Now, can this workflow be done on other matrices besides beef, chicken, or pork muscle?
Okay, yeah. So as we presented this, the data is for 3 matrices, which is chicken, beef, and pork. We have verified the workflow for these 3 matrices. The method can be extended for other matrices, but right now it's not verified. But the protocol is definitely can be extended for other matrices. Okay, one more thing is like we are in the process of extending this workflow for other matrices, and we will release the data with other matrices in up north in the near future.
Thank you, Siji. Now, gentlemen, can you share what are the advantages of Captiva EMR lipid extraction if compared with DSP?
Okay. Captiva, as I mentioned during my presentation, is very easy to use. It's like almost like a hands-free operation once we load the eluents, the extracts to the cartridge bed. And it's designed for gravity elution, and all the lipids will stick to the bed, and the analytes will freely pass through the bed, and it's easy to recover that way. And the Agilent PPM is a kind of, we can process 48 samples simultaneously in less than 30 minutes. So it's all very user-friendly compared to the conventional DSP technique. We don't have to do any of the steps like preconditioning, conditioning, or washing kind of steps in the Captiva cleanup procedure.
Thank you, Siji. Now, gentlemen, I'd like to go with this question. It's come in a few times. Could you elaborate on the application service?
Patrick?
Sure, I can take this question. Yeah. The application service is it's another component to the overall package of this workflow. And what it is that you can get consultation service that helps you successfully execute this workflow and navigate through any bumps or issues along the way. So this is essentially, as you practice this workflow and incorporate this into the lab, you have the watchful eye of a professional Agilent applications chemist, just so that you can catch any pitfalls, help you through the software, help you customize your reporting, or anything like that. Just to make sure that your transition, from a conventional workflow to something like this is very, very smooth.
Thank you, Patrick. And gentlemen, we have time for a few more questions, but I do want to remind our audience that any questions we're unable to answer today due to time, they will be addressed by our speakers via the email address you provided at the time of registration. Okay, let's continue with our questions. 2 LC-MS triple quads were mentioned in your talk. Which instrument do you ultimately recommend for this workflow?
I'll take this question. In regards to the instrumentation, we recommend a Agilent 6470B triple quadrupole LC-MS or a 6495C triple quadrupole LC-MS. Ultimately, the workflow was designed for and to hit targets that are achievable by the 6470B triple quadrupole. However, if you wanted that extra confidence or that extra sensitivity, you can always use the 6495C triple quadrupole. So to answer that question again directly, the workflow was designed for the 6470B, but can run on the 6495C.
Thank you, Patrick. And with this workflow product, what is the cost per sample?
Yeah, I'll take this. So we have a kind of estimated the use of consumables for executing this for the daily run, and it's about per analysis, we can say that it's about $0.5 is the cost of analysis. Thank you.
Thank you, Siji. Can laboratories use this workflow solution as a validated SOP?
Okay, that's a great question. Yeah, we are promising a verified method, and still customers need to verify or maybe create an SOP as per their local laboratory need. Definitely, we have verified it. You can use the methodology and the results to build your own SOP to meet your laboratory need. But the short answer is like the customers needs to re-verify at their laboratory using this as a fundamental basic results or methods to build their SOP.
Thank you. And what is this special dynamic MRM mode, and what are its benefits?
Sure, I can take this. The dynamic MRM is something that maximizes the instrument cycle time for maximum sensitivity. So it's essentially pointing the instrument to analyze a specific analyte at a specific point in time. And the reason why we state that it is dynamic is that the user indicates a retention time an analyte will elute at, and the instrument will automatically adjust and account for the overall cycle time. So if an analyte is not eluting at that specific time point, the instrument will not acquire that MRM during that duty cycle. So as the analyte starts to elute, the instrument points out to only what's relevant, and this thereby increases the precision and also the sensitivity as well.
Thank you, Patrick. And how is this workflow solution approach better than the conventional approach that many labs are doing now?
I'll take that. Yeah, so this is like the workflow solution is like a single vendor solution which includes various components to successfully implement and perform the workflow in a customer laboratory. This includes workflow, sample preparation, the LC-MS method, data analysis procedures, and also reporting templates. So it's a package, and very importantly, even the consumables also. So of course, you can build it or you can develop a method from scratch. But again, as everyone knows that method development is one of the most time-consuming activity in any analytical situation. So this workflow solution definitely helps a customer to start or implement a workflow solution from the day one and start analyzing the samples from the day one, because it's already ready-made and everything is like a single vendor solution. Hope that answers.
Thank you. And we're going to have to wrap with this final question. Can you elaborate more on how to use the dynamic MRM database when creating a new method in the lab?
Great question. Thank you. Yeah. So this is like the MRM database includes of MS parameters like the product ions and the mass specific parameters for all 210 targets. And a customer can search their preferred targets from this database using search keys, like they can search for CAS numbers, or they can search for regulatory specific, like EU specific or US specific kind of targets. So we are also providing a method which is a kind of base method, and customers can very easily import the relevant interested targets to this base method and save into their subset method. So it's very easy, and we have given all the instructions in the workflow guide, which is included in this workflow solution. Thank
you, Siji. And gentlemen, thank you both. I'd like to big thank you to Dr. Patrick Batoon and Siji Joseph for their time today and for their important research. And before we go, I do want to thank our audience for joining us today and for their interesting questions. We had quite a few questions we were unable to answer today, so as a reminder, those questions will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand through June of 2021. Thank you for joining us. We hope to see you again soon. Have a great day.